Hello, everyone. Good morning, good afternoon, good evening, depending on wherever you are. So, I would like to begin by thanking the organizers for this wonderful opportunity of sharing some snapshots of the exciting population genetics work that I have been associated to by virtue of being a part of the H3 Africa Consortium. So, let us start with a simple question. How different is my or say your genome from the 3 gigabase reference human genome that was published in 2001? So although the exact number would probably vary, but on an average a genome differs at 4 to 5 million sites. That's a A will be replaced by a G or a T or an insertion or a deletion from the reference genome. So therefore, in terms of percentage, less than 0.02% of the genome that we would normally sequence will be different, while the rest of the 99.8% plus would be exactly the same as the reference genome. However, as you know that it is this small percentage that contains all the critical information, such as what will be the color of our skin, eye, whether we will have predisposition to certain diseases, whether we will be able to digest milk when adults, or we will be able to be benefited by particular medicine, and so on. But what is more interesting to us as population geneticists, that in addition to all this information, a genome is also kind of a time capsule. So it condenses the evolutionary history of all the ancestors and can provide answers to questions about them, such as whether they migrated or not, whether they made and exchanged genes with other populations, what was their population size through time, and also gives us some idea on the diseases and pathogens they might have encountered. However, a single genome can only provide limited information. As you can see in this plot, each of us harbor a slightly different set of variants. So the moment you bring in more genomes, the dataset size expands, and with about 100 individuals, you can capture variations at around 0.6% of the genome, and this can give you quite reliable information on many aspects of evolutionary history. So as a comparison, while a single or a couple of genome gives you a low resolution picture, with population scale data, let's say 20, 50 or 100, you start getting higher resolution pictures, and the more genomes you sequence, higher is the resolution of evolutionary history that you get. As you can see that the number of variants detected also shows quite a wide range of variation. So for an individual it varies between 4 to 5 million, whereas at the population level it varies between 17 to 23 million. So what is the reason for this variation? So this variation is actually due to differences in ancestry of the particular individual or of the population. So as this plot from the 1000 Genomes Project study shows, here the number of SNPs in each individual is represented by a dot, and the same population is shown in dots of same color. African genomes on an average harbor almost 25% more variants compared to non-Africans. So why do Africans harbor so many more variants? So this figure from Karina Slebush's 2017 paper demonstrates why this happens. So as you will note here, all the initial divisions, that's the split of ancestors of Khoisan hunter-gatherers, followed by the divergence of ancestors of rainforest hunter-gatherers, and here you can see the y-axis actually is time, so there you can see that some of the splits are really ancient. So finally the split of the East and West Africans, which is also probably around 100,000 years old, all happened within the continent, and it is estimated that all the current non-Africans are related to the East African branch, as the out of Africa migrations originated from this. Therefore, barring this, all branches and all other ancient lineages remained and flourished only in specific regions of Africa, which enabled them to be so diverse. And despite this immense diversity, African genomes are so far the least studied among all populations. It's very difficult to estimate these numbers, but a reasonable estimate suggests that of the first 100,000 genomes that have been sequenced, less than 5% were Africans. And although projects such as StopMed are trying to bridge this gap, the genomes included in these studies are predominantly from African diaspora. And as we know from our knowledge of history, these diaspora populations represent only certain African geographies and ancestries, so they are not completely representatives. And even among the genomes that were sampled from the continent, as the slightly dated map of population scale WGS datasets depict, there is a clear bias towards certain geographic regions and huge gaps on the map. And only a few of the 2,000 or so ethno-linguistic groups have any representative genomic data. So there is definitely a lot missing here. And the scenario is equally or even more bleak if we look into disease genetics data. Despite continental Africans comprising about 15% of the world population, less than 1% of participants in disease genetic studies are continental Africans. So the genesis of the studies that I'm going to talk about today lies in the Human Heredity and Health in Africa, or H3 Africa Consortium's decision to do something about this gap. So for those who are not familiar, H3 Africa is a National Institute of Health, Wellcome Trust, and African Academy of Science funded project to establish genomic tech and expertise across Africa. This is a $180 million initiative involving 50 projects, 500 researchers, and spread across 34 African countries. So today I would start with how our WGS study informed our understanding of diversity, selection, and migration. And as many of you know, this was published in Nature last October. And given the current standards of whole genome sequencing projects, a study based on 426 genomes is indeed quite modest. However, what makes this study special and perhaps enabled us to garner so much attention was the fact that the reported whole genomes were from Africa. So the geographic spread of the consortium that we saw a couple of slides ago enabled us to strategically prioritize 323 samples from 8 countries, which could fill in some of the major gaps. And these samples were sequenced at 30x or higher depth. We also included 212 individuals from 4 other countries that were sequenced at medium coverage by another project within the consortium. And after quality control, we ended up with a data set that consisted of 40 million variants and 426 individuals. And this represents 50 different ethno-linguistic groups. So among the 40 million variants that we identified in our study, almost 8%, that's 3.4 million variants, were novel with respect to variant databases and previous sequencing efforts. So this was indeed quite high. And to unpack how ancestry and geography are reflected in the novel SNV content, we compared the novel SNV counts normalized to 24 individuals per population groups, as you can see in this plot. And although genomically speaking, Nigeria is the best studied African country, the Berome from this country, which is shown here, was found to have a large proportion of novel SNVs. And this kind of emphasizes that the current Nigerian groups are not only poor proxies for the whole of Africa, but maybe poor proxies for Nigeria as a country as well. However, the novel SNV represented here was pale in comparison to what we saw in Botswana. And admixture plots for Southern Africa show that the Botswana have significantly high Khoisan ancestry, here shown in green. And we also found a strong correlation between the proportion of the Khoisan ancestry and the number of novel variants in an individual. So similar correlations were also observed for other ancestries such as North Africans, rainforest hunter-gatherers, and so on. And this kind of shows that there is a tremendous potential for novel variant discovery in African populations if the populations are chosen so as to bridge geographic or ancestral ancestral representation gaps. So we then surveyed the frequencies of select well-described disease-associated variants, and some of these are depicted in this picture. So RS334, which is the common sickle cell disease mutation locus that protects against malaria mortality, was found at typically high allele frequencies in malaria endemic West and East African populations, and this was expected. However, what was interesting was that the HBS showed widely divergent allele frequencies in two populations from Uganda, with the Bantu-speaking UBS population having one of the highest frequencies, that's around 20%, whereas this allele was not observed at all in the Ugandan Nilo-Saharan population. And while this was due to major ancestral divergences, as these PC and admixture plots show, these extreme differences underline a critical point that for Africa, estimates of disease susceptibility or genetic risk needs to be much more fine-grained, and a single estimate for a country might often be misleading. So next, we tried to assess the contribution of natural selection to the genomic landscape. And for this, we employed a very widely used estimate known as composite likelihood ratio, or CLR, in each of the population. And this method is actually, using frequency of mutations in a genomic region and using a quite complex computational methodology. It assesses whether a region has undergone selection or not. And this circular Manhattan plot shows the selection signals, or CLR scores, across 10 kb windows, and this is like the whole genome, and the concentric circles are for Botswana, Zambia, Cameroon, Nigeria, Benin, and Ghana and Burkina Faso, moving from outside to inside. As this is quite difficult to see, you have to take my word for it that we identified 107 signals in these 6 populations, and 62 of these were in novel genes. And as expected, a large proportion of the novel genes were involved in immunity, DNA repair, and metabolism. So immune genes are somewhat common in any selection scan, and several signals for immunity to protozoan diseases such as APOL1 and HBS are very well known in African populations. However, what was surprising was that a substantial proportion of immune genes detected in our scan were related to viral immunity. And this suggests that viruses might have also played a role in shaping genomes, at least in some parts of Africa. And although almost half of these novel signals were seen in 2 or more populations, several were seen, as you can see in this rectangle, that they were either population or geographic region specific. Moreover, for some of the well-known signals such as FOXP2 and APOL1, they showed very high variability in selection strength across the groups studied. So given a recent shared history and geographic proximity of some of these populations, these differences were intriguing, and it would be a great topic for in-depth investigation as larger WGS datasets become available. So moving to migration, so long distance migrations have played a pivotal role in shaping the genomic landscape of the African continent, especially in the last few thousand years. And this is mainly by bringing ancestral populations inhabiting different regions of the continent in contact with each other and resulting in this characteristic distribution of linguistic groups. And there are several important migrations that shape African diversity. The most dramatic of this is perhaps the Bantu expansion, a series of migration that initiated around 4,000 years ago from somewhere in modern-day Nigeria and Cameroon to vast areas across sub-Saharan Africa. And it is due to this migration that one of the Bantu family languages are the major languages of almost all sub-Saharan African countries. However, the exact sequence, timeline, and route of this migration is on one of the most debated topics in African archaeology, linguistics, and perhaps also genetics. So the PCA of our data, that when it was integrated with data from external sequencing studies, looks like this. And as has been observed for similar populations in previous studies, the first principal component, which is along the x-axis, separated the East Africans from other African populations. The second principal component placed the remaining populations along a line from west to south, and it's kind of like shows a very strong correlation with geography. So the individuals from Cameroon, DRC, and Zambia localized in proximity to each other and forms a somewhat independent Central African group. And here you can see the populations from Zambia kind of localizes in the crux of this line from west to central and then to south and east. And this unique localization of this group in the PC made us suspect that it might have interesting connections with other neighboring groups and perhaps maybe to Bantu migration as well. So the most current genetic database model is from a study by Patan and colleagues published in 2017, and it estimates a southerly movement from Cameroon to Angola before dispersal into south and east. And the inclusion of populations from DRC and Zambia provided us the opportunity to explore the potential role of these countries in the Bantu migration route. So when we looked at sharing of shared genomic segments, that's IBD sharing, we saw that the sharing with Southern African Botswana was greatest among the Zambians, and again all the East African Bantu speakers from Uganda were also found to show highest genetic affinity with Zambia among all the Central and South African populations. And we performed several additional analyses using admixture modeling, and all of them support the view that Bantu migration moved south of rainforest to present-day Angola and then perhaps moved eastward, and only after reaching Zambia the eastern and the southern streams separated out. So as you can see that this study has a lot of population genetic insights, and there was another utility which this genetic data kind of served. And as we all know that despite the plummeting cost of sequencing, the relatively cheaper price, ease in data handling and storage still makes genotyping array very widely used in disease genetic studies. And also much of what we know about population genetics comes from array-based studies. However, arrays designed on the basis of European datasets was long known to perform suboptimally in African populations. And as the major aim of the H3 Africa was to conduct GWASs, the consortium decided to design its own Africa-centric array, and the novel genomes along with public datasets contributed to the design. So a key feature of this array was that in addition to 1.5 million SNPs from existing libraries, which are called beat pools, the array contained around 700,000 SNPs that were specifically inserted to better represent African LD, and there were a lot of functional variants which were also targeted to capture the SNPs that might be relevant for African GWASs and other studies. So this array has been used to genotype more than 50,000 Africans to date. And in the second part, we will visit a recent study that was based on data that was genotyped on this array, and we will see how it enabled us insights into the final stages of Bantu migration and the peopling of Southern Africa. So going back to Bantu migration, as the migration moved south of Zambia, something very interesting happened. So the migrants came in contact with Khoisan hunter-gatherers which represents one of the most ancient and genetically diverse human lineages. And this was followed by a gene flow from Khoisan into Southern African Bantu speakers, which makes them very unique and perhaps uniquely diverse among all Bantu speaking populations. So the Bantu speakers just from South Africa speak several distinctive languages such as Zulu, Swana, Xhosa, as you can see in this tree. However, whether these linguistic differences are paralleled by genetic differences was not known. And although one of our earlier work hinted at this possibility, it was a very limited study based on a few samples and just two ethno-linguistic groups, so nothing was conclusive. So employing the Avigence dataset that included 5,000 participants from all major South African ethno-linguistic groups, we investigated whether there is a correspondence between language and genetics. So as you can see, the distribution of 9 major ethno-linguistic groups on the map as well as the language tree. Now the genetic data shows, as this you can see in this PC, a separation among the 3 major ethno-linguistic divisions, that's Sotho, Swana, Nguni speakers and Tsonga. And not only that, if you look carefully, you would see there is a correlation between localization of these groups on the map and their localization on the PCA, showing that there is not only a population structure within the ACB, but the structure correlates both with language and geography. So key components of the history of a population are often reflected in how the size of the population has changed. For example, a population that's affected by adverse climate change or a disease would have undergone a reduction in population size. On the other hand, exposure to new resources or cultural changes such as adoption of a new farming technique could increase the population size dramatically. So we used a program IBDNE, which based on this premise, and it tries to investigate the changes in effective population sizes in last few hundred generations. So in this plot, we have summarized the population size changes in ACB groups in the last 100 generations. And as you can see in this figure, that all ACB groups are very similar for the initial 60 generations, and only then the individual groups began to diversify. And this matches with the generally accepted model of Bantu expansion, which suggests a sharing of a common ancestor by all present ACB groups until this time range. And any variation profile of the Nguni speaker, that's Zulu and Xhosa, was found to be similar but different from all other ACB groups. Similarly, the Sotho Swana also kind of shows a distinctive and characteristic profile, suggesting that the population structure that we observed has some level of correspondence to demographic history. So to delineate the timeline of the migration of Bantu speakers within Southern Africa, we dated the admixture between Bantu speakers and case source populations for each of the ACB groups using various techniques. So in this plot, dates based on fast globetrotter are shown in red, and those based on Mulder are shown in blue. And our dates show an overall correspondence with previous studies, but provides a more granular estimate of these admixtures. And the Tsonga shows the oldest admixture dates, which is around 45 generations ago, while admixture dates for other groups such as Swana and Xhosa were around 25 generations ago. And these dates are supported by archaeological evidences which indicate that Tsonga might have indeed arrived before the Nguni speakers. So this kind of shows a model of Bantu speaker expansion within Southern Africa to be a complex process which could have either involved multiple waves of migration or some form of punctuated expansion. So as we performed this analysis for South Africa, for this review, we tried to expand it, this kind of estimates for a wider geographic region. And for this, we just used available literature and we faced a major challenge. As you can see here, each study out there has a different genotyping array that was employed. There were differences in dating methods and the Khoisan proxy data sets. So all these factors can affect the estimates of admixture dates. So we have a situation where we have substantial data to provide a decent timelines. However, as these studies were conducted in kind of methodological silos, unless we integrate them and homogenize them, we cannot do that reliably. So the collaboration within African researchers is required to use datasets in such a way so that we can homogenize them and generate both Pan-African and regional insights. So to summarize, the fruits of H3 Africa Consortium can be broadly divided into three categories. Firstly, to bridge the gaps in publicly available whole genome data, we have been strategically sequencing hundreds of whole genomes at both high and medium coverage. So some of these are presented in this talk and many of them are to be published soon. The consortium based on this data has generated an Africa-centric genotyping array, and we are busy integrating genomes into an imputation panel that would enable better imputation of continental African data sets. And the data generated using H3 Africa genotyping array has not only enhanced our understanding of complex diseases by means of GWAS, but also leading to major leaps in our understanding of population structure and demographic history of specific African regions. And all these data sets are being made available via global repositories such as EGA, as well as local databases aimed at enabling African geneticists and bioinformaticists to be able to make use of these for their own research. So finally, I need to emphasize that I'm presenting all this work on behalf of three wonderful teams, and all the credit goes to these teams. And finally, sincere thanks to the funders who made all this possible, SBIMB, my research home, and the organizers for this opportunity, and all of you for your kind attention. Thank you.